so much for having us, and was the final talk in this month's Science in the News, again put on by Yale Science Communication. I'm Nick Fisk, a fourth-year computational biology PhD candidate at Yale in the Townsend Lab. My main role here tonight is to introduce our fabulous speakers and their talks. Speaking of, here they are. So we're going to talk just a little bit about the speakers today, show you what cool people they are. First up, we're going to hear from Shannon, who's in Yale's Biological and Biomedical Sciences. Her research focuses on gamete and early embryo epigenetics, and she has performed with a dropkick Murphy. So if you're anything like me, you'll need some clarification in that the dropkick Murphys are indeed a band and not an underground MMA ring. Then we're going to hear from Damir, who's in Yale's Department of Genetics. His research centers around mRNA biology and embryonic development. And a fun fact about Damir is that he plays guitar and was formerly in the Yale Russian chorus. And rounding us off for tonight is Hadass, who's also in the Yale Department of Genetics. And her research focuses on neurogenetics and neurodegeneration. And a fun fact about Hadass is that she served in the Israeli military and has a pet axolotl. So in order to prime you a little bit for these lovely talks, I'm going to introduce some key concepts to you all here. And I want to do so by sharing something that I find utterly fascinating, even after 4 years in a PhD program, and that is how complex and intertangled the different systems in the human body are. So of course we have the skin, which sort of holds us in and keeps us safe from all the outside germs and problems of the world. And you have the musculature, which lets you move around and interact with the world in meaningful ways, protect yourself and others. You have your skeleton, which serves as this really nice foundation for your whole body to attach to. Your nervous system, which actually sends the messages to the rest of your body, even subconsciously, to keep your organs functioning properly. And of course, we have the circulatory system, which brings oxygen and other nutrients to different parts of your body that need it. And what always astonishes me, and I still can't get over, is the fact that no matter who you are, all of this starts as one cell. Right, so all of that information to make you and make all these different systems, they start just from one cell, just some cell and some time, and you end up with all of this vast complexity. And this process is quite beautiful and also incredibly complex, and scientists still don't know everything about it, leaving it an incredibly rich field to talk about, like we will tonight. And so I want to talk a little bit about how that one cell can become many cells. And that one cell is usually formed, as you'll hear from Shannon, when a sperm and an egg meet and create something called a zygote. And then through time, that zygote divides and replicates and creates more cells. And as time goes on, some of those cells become more and more what we call specialized or differentiated. So some of them will end up as muscle cells, some will end up as bone cells, and all the other cells in your body. And a cell who has that property to become other things, we tend to call stem cells, right? So that property, you often hear scientists refer to them as stemness, right? And that is just simply the ability to become more specialized types of cells. So a stem cell hasn't quite decided what job it wants yet, and you can think of it kind of like the smiley face, where you can kind of have different flavors of smiley faces, right? These emoji. And in a lot of ways, then a zygote, that is what happens when your sperm and the egg mix together. You can think of those as like the ultimate stem cell. They can still produce any cell in the body. And how can they do that? And how do stem cells sort of work is something we'll hear a little bit about tonight. But I broadly want to bring up the idea that all of the cells in your body, including this one zygote cell at the beginning, they all have the same information in them. And that information is stored long-term in the form of DNA, which you may have heard of. And what differentiates the different cells in your body from one another, from the skin, from the muscle, the skeletons, and more, have to do with what recipes in the DNA each cell decides to use. So the different recipes they'll have and how much of that recipe they'll make. That's ultimately what distinguishes the cells from one another. So even though all the cells in the body have the same information, they don't all use it in the same way. And so that's an incredibly fascinating facet of how cells sort of become from this, very high level stem cell all the way down to your body parts today, which we'll hear a little bit from Damir and others. So without further ado, I want to introduce our speakers and their talks. First will be Shannon talking about conception and paternal and maternal information, how all the information that ultimately becomes and is used to construct you, how does that get into one place? Then we're going to hear from Damir, who's going to actually talk a lot about embryonic development, how different parts of your body know what to be. How do your hands know to be hands? How does your brain know to be a brain? And how do we know how they know? And then lastly, we're going to end the night off right with Hadass, who's going to talk to us about aging, both how and why we study aging, but also how we are learning to age well. And so without any further ado, please welcome Shannon to give us our first talk.
Awesome. Thank you so much for the introduction, Nick. Let me just pull up my presentation. And as Nick was saying, I'll be talking to you guys a little bit about conception, or this idea that this information flows from our maternal and paternal side all the way down to us. And ultimately, I want to answer the question of, how did we get here? And so a lot of kids ask this question when they're younger, and people will talk about these birds and these bees, or some parents will talk about how storks come along and bring babies down and deliver them onto people's front doorstep. But when my sister and I asked this question, my mom gave us this book called It's So Amazing. And it's a book that covers all this information about what eggs and sperm are, and is also about how we get all this information from our parents to us, and how we inherit different kinds of things from each of our parents. And funnily enough, the book features both a bird and a bee. So I want to talk a little bit about why I was so fascinated with how we got all this information, and answer a couple questions during this talk. So the first question that I want to answer is, what does this information look like in people? And so I will talk a little bit about books, libraries, and cookies, which I promise by the end will make so much more sense, and hopefully not get people too hungry. And then the second question I want to answer is, how exactly did we get here? And what is this physical flow of information? And so I'll talk about the process of fertilization. But to start, let's think about information and what does it look like inside of us? So I've always liked the analogy that people are like libraries. We are chock full of all this vast information, and people are full of something called cells, just like libraries are full of books. And so we can think of cells almost like a recipe book. It holds all the information that's encoded to help make us who we are. And so over here, we have a picture of what a cell looks like. And this information that I keep on talking about and repeating about is localized inside of this thing called the nucleus, which almost acts like the brain of the cell. And so if we are like libraries and our cells are like recipe books, then the information that's inside must be each individual recipe. And so what's inside of a nucleus is something called a chromosome. And so in this analogy, again, we can think of these chromosomes like each individual recipe. But what exactly is a chromosome anyway, and why do we even care about them? Well, each chromosome holds all the information that will ultimately make us who we are. And so here in this picture, I have something called a karyotype, which some scientists and doctors will use in order to look at each child's chromosomes. And so, so we have here is the nucleus of a cell, which has all of these different colors. And scientists can stain each individual chromosome to have different colors and ultimately pull them out into these tiny little worm-like things. And this is exactly what a chromosome looks like inside of one cell, inside of all the cells that make us who we are. And a really interesting thing about these chromosomes is that we get one of them from each parent. And so over here in the insert, as you can see, you can pair up each of these chromosomes into 23 different pairs. And you ultimately get one of the pairs. So for example, on this chromosome 1, this left side could belong to the mother, and this the right side could belong to the father. And so these chromosomes ultimately pass the information on from our parents to each and every one of us. So what's inside of these chromosomes? So inside is something called DNA. And so we can use DNA and chromosomes kind of interchangeably to think about these recipes. And so if we have a recipe for something and we wanted to express it, that is exactly what a chromosome and each of the genes or traits inside of us do. So they hold this information for something called a trait. A trait is something that is usually physically expressed inside of a person. And so, for example, I have freckles, and these freckles come from a gene which I inherited from my mom, who also has freckles. And so, what exactly are these traits? How do they come to be? What is the process that actually moves this DNA all the way into something that is expressible? And so we can talk a little bit about how this recipe is expressed inside of each of the cells. So we can think of DNA or chromosomes like these chocolate chip cookie recipes. Well, each of the cells can make a copy of this recipe, almost like a recipe card. And so this is called RNA inside of the body. And so RNA is like a copy of the original recipe. And so these cells can take these copies of the recipes and ultimately make the recipe, like chocolate chip cookies. And so the expression of this copy or translating it into something that you can eat is similar to how a cell is able to translate RNA into something called a protein. And these proteins ultimately encode the different kinds of things that will make up our traits. And so takeaway from here is that we have these DNA, which almost acts like a recipe. We have a copy of this recipe inside of us called RNA, which ultimately makes something like chocolate chip cookies, which is known as proteins inside of us. And so all this basic biology can help us understand a little bit more about how we get here and how this physical flow of information actually gets to each and every one of us. And so I talked a little bit before about chromosomes and how we get one of them from our parents, each of our parents. And so how does this actually look at the beginning, earliest stages? So in the mom side, we have something called an egg. And an egg has one copy of each of the 23 chromosomes. You can combine that with the father side, which is sperm, which also has 23 chromosomes coming from the dad. And so when you mix this egg, which has the mom side, with the dad, which has with a sperm, which has the dad side, you come up with something called a zygote. And as Nick has mentioned, a zygote is the first single cell that will ultimately divide in order to make all the different parts of what you are. And so inside of the zygote, you can see that there are 46 individual chromosomes, or 23 pairs, of which you get one from each of your parents. And so this whole physical flow of information happens quite literally. And so we can think of this kind of flow starting off on the dad side. So we can ask ourselves, how exactly does this sperm meet an egg? What is this physical flow? So sperm, let's talk a little bit about them. They are produced inside of the male tract in this area called the testes. And Nick had mentioned a little bit about stem cells, and inside of these testes, males also have stem cells. These stem cells, or spermatogonial stem cells as they're known in this area, are able to divide and differentiate and change into a sperm. This whole process is called spermatogenesis. And so males have a bunch of different spermatogonial stem cells that will keep on renewing themselves and constantly be making sperm. So after these sperm are created inside of the testes, they flow to this area called the epididymis. And this is where they are stored until they enter the female tract, and they also gain the ability to move at this point. And so here I have a little GIF about what sperm actually looks like, which is absolutely wild because it is such a small size. So here we can see a couple of sperm moving across the screen. They have a head here, as you can see at the top, and then they have a spinning, flowing tail on the back, which propels them forward. And so we can think of these sperm like little self-driving vehicles of information from the father side that ultimately need to make it to the mother side, combine together and make this one single cell, which will eventually divide into all of us. And this whole race starts off with this sperm and an egg. And so it's absolutely wild to think that each and every one of us came from the combination of these two parts. And this whole race, which we talk about as the Great Sperm Race. And so this whole race starts off with the sperm coming into the female tract, flowing through the cervix, going through the uterus, and then ultimately finishing in one of the fallopian tubes. And so at the beginning of this race, which makes each and every one of us, about 250 million sperm will enter the tract. But they are met with a bunch of different obstacles at first, which include the acidic lining of the female tract, along with the female immune response. And both of these things will combine together and kill off a ton of sperm so that only 1 in 10 will actually make it to the end, or about 2 million will be able to flow to the next stage. And so you think to yourself that these sperm are having a hard time, like they got to have a little bit of like a leeway or some kind of easy part next, but they don't. So about 2 million sperm will enter this area called the cervix. And the cervix is almost like a maze. There are a bunch of different crypts and areas that a sperm can get lost in within the cervix. And it must choose the right path in order to make it to the end. And so within the cervix, about 1 million sperm will get lost, which means only half or 1 million will be able to make it to the end. And so after they flow through the cervix, they enter the uterus. And so the uterus is almost like this vast void, and the sperm doesn't quite really know what to do. Sperm can either go straight through and kind of get lost, or it can shoot up through the sides of the uterus wall. And then another issue that sperm ultimately have to make is the decision as to whether or not they want to go left or right, because at least in the humans or some mammals, only one side will ovulate at a time. And so an egg might only wind up in one of these fallopian tubes. And so with all of the luck that they have, it is possible that only 10 sperm will actually make it to an egg at any given chance. And so finally, after going through all these obstacles, a sperm is able to meet an egg. And so here we have a video of what an egg is. You can see the oocyte or the egg is actually darker. It has a shell around it. And down here in this insert, you can see a single sperm breaking through the shell in order to make it to the egg. And this is the exact moment of fertilization. This is the moment when the father's information, chromosomes, are able to meet the mother's chromosomes or information, combine together in order to form something called a zygote. And so this zygote is the first single stage that will continuously divide again to make each and every one of us. And so it's crazy that all this comes down to some kind of race by some kind of driverless car, basically. And so just an overview about what I've talked about so far is that I really want to reiterate, it is so amazing, this whole process that we're able to get here, that this tiny packet of information can reach this other tiny packet of information in order to create everything of who we are. And that we are ultimately like libraries. We are full of these things of cells, and these cells have chromosomes and DNA, which ultimately express these different traits that we have. And that this great sperm race is something that is one of the greatest moments of our lives that honestly none of us remember. And so this whole process of a sperm getting to an egg is ultimately the first stages in order to create our lives. And so once a sperm reaches an egg, it is fertilized to a zygote. It divides a couple times all the way up till this blastocyst stage. And then after that, one of the questions that we have is, how does this mass of cells know what is its hand, what is its head, what is its brain? And to talk a little bit more about how that whole process of differentiation and gastrulation happens, we can talk to Demir.
Thank you, Shannon, for very nice introduction on fertilization and the first stage of our lives. So, hi everybody, my name is Damir, and I'm going to talk about how the different parts of the embryo form. So, as you learned from the first talk from Shannon, the first stage of our life is actually this process called the fertilization, when the egg and sperm meet. And it is incredible and it's fascinating how this single fertilized cell eventually over time transforms itself into this beautiful embryo with its head and eyes and limbs already developed. And one of the big questions in the field of biology is to actually trying to understand this process. How do we get a complex organism from a single cell? And how does it know what to become and what shape to acquire eventually? And this particular question has been in our minds for ages. Even in the 4th century BC, the Greek philosopher Aristotle was addressing this problem of how do different parts of embryo form. And he proposed 2 hypotheses. The first hypothesis was that everything simply gets bigger over time before it is born. So what he meant is that everybody starts off as a little human being inside of the womb, which eventually keeps growing and growing and growing when it reaches certain size, when it's ready to be born. And the second hypothesis was that new structures arise progressively. And what he meant by that is that we start off by being in a certain shape and size. Eventually, over time, we change to different forms and shapes, eventually giving rise to a complete embryo, a complete baby before it is born. So we had these 2 schools of thoughts about that everything gets bigger over time or new structures arise progressively. And let's try to think about these 2 possibilities. So possibility number 1, even in 17th century, people believed that each sperm has a little person in its head, curled up in its head, and they called this little person a homunculus. So what they thought is that this little person inside of each sperm would be delivered into the womb, where he or she will eventually grow before they are born. However, this hypothesis was completely abolished, and the view has changed when it was recognized that all living things, including embryos, were composed of cells. And here's a representation of the human cell under the microscope. This is a human skin cell. And so, as you can see, like by the borders, this is the cell membrane, and the dark spots are the nucleus, where we store all of our DNA and all the chromosomes that Shannon introduced at the beginning. So scientists recognize that even embryos compared to adult human being have relatively the same size of the cells. So it was very hard to imagine that there would be a little human being curled up inside of sperm. So the hypothesis number one was rejected, the one that says that everything gets bigger over time. And people started to think about the second possibility, that the new structures arise progressively, that the shape and the form changes over time and before it is born. And people were trying to understand the process, as I told you at the beginning. How do we get from a single cell to a beautifully organized embryo with all the organs and all the parts already formed? Before I tell you what we learned so far over the past hundreds of years, I want to ask you one question. Which one of these pictures illustrated here you think is a human embryo? Is it number one? Is it number two, number three, four, or five? You can put your answers in a chat, or you can make a note for yourself. Try to guess which one of these you think is a human embryo. I'll give you some time, take your time. This is how the human embryo will look around 4 weeks after fertilization. Is human embryo number two? Is human embryo number 4, 5, 3, 1? Which one do you think? Okay, I think we're getting different answers. 3, 4, 3, 5, all 3, 5. Good. So interestingly, the human embryo is embryo number 5. And number 4 was a mice, number 3 is a chicken, and number 2 is fish, number 1 is a frog. So what you can appreciate is that at the beginning, at the early stages of development, the general body plan more or less looks very similar. So each embryo has its head, its eyes, its muscles, its limbs starting to develop, and its brain is starting to grow. So, and the scientists were really trying to understand, how do we get from a single cell to this structure even after 4 weeks? So, of course, for the ethical reasons and for convenience reasons, we cannot study this process in human embryos. And when I say for convenience, I mean just the size of the fertilized egg in human embryo is way, way smaller compared to, for example, the fertilized egg of a fish or fertilized egg of a frog. So it would be very easier for scientists to use these model organisms to really understand the development of embryos, because they will be easier to manipulate, easier to observe under the microscopes. So actually, understanding the developmental process in one organism can help us to eliminate similar processes in another one. So many of the developmental biology questions were asked using the frogs. And the life cycle of frogs starts with fertilization, when the sperm meets an egg. And eventually, after a couple of hours, these one cell keeps dividing, giving rise to like hundreds of cells, where we have like this bag of cells called blastula. And after several rounds of division and cell migration, we get this beautiful embryo with its eyes and head and muscles already formed. And the question is, how do we get from this bag of cells, like hundreds of cells, to this embryo with most of the structures already formed? And I'm going to give you the answer right away. It seems like even when you look at this bag of cells, like hundreds of cells, they already know what to become. So what I mean by that is that, so the cells on top, they already determined that they want to be skin cells. The cells in the back determined to be brain cells, and so on. So the body plan has been created early on, even though the cells have not acquired any shape or form yet. So let's try to go through it one by one. So I'm just trying to represent this blastula stage as these orange cells. So each orange circle here is a cell. And what I told you so far is that there are certain cells in some part of the embryo determined to be, for example, brain cells. So these cells, they try to stick with each other and they say, we want to be a brain cell. We want to give rise to brain structure and the head. And while the other cells, they also try to stick with each other and they said, oh, we want to be a muscle cell. We want to make this embryo strong. We want to find the relative position where we will be eventually muscle cells everywhere. And what scientists learned is that the fate map is created early on, even though we have this bag of cells that give rise to the head and tail and all the parts of the body. These particular cells act like I don't know, like a GPS. They position themselves in a relative to each other in a way so that they know what to become. When the skin cell eventually keeps dividing and growing, it will actually give rise to a skin cells that will cover up the whole embryo. And the same for the muscle cells, it will give rise to a muscle structure by migrating and growing. And you might be asking, how do we know this information? Like, how did scientists figure out that embryos have determined their fate early on? So it's a series of interesting experiments scientists have done. I'm going to show you a couple. So here, the scientists took two embryos, so the embryo on the left and the embryo on the right, so two bag of cells simply, and one is a donor embryo, the other one is a recipient embryo. So they took the cells that have already determined their fate to become brain cells, and they transplanted these cells from a donor embryo to a recipient embryo. Now the recipient embryo has two sides with cells that want to be a brain cells. So the question is, how do you think this recipient embryo will develop? Take your time, try to think about it. What do you think will happen? Would these embryos on the right, would they die, or would they give rise to two heads, one head? Or would these cells that were transplanted, they will be rejected? Or what do you think will happen to the recipient embryo? I'll give you a couple of seconds to think. So simply what I said, we transplanted the brain cells from one embryo to another embryo. And it's the early stages of development. We still have this bag of cells. What do you think will happen? Migrate to another cells, die, two brains. So those are good guesses. So interestingly, when scientists did that experiment, when the cells were transplanted, they saw that embryo developed with two heads. So here's a picture of the two-headed tadpole. So what it means is that if the cell determined its fate, if it wants to be a brain cell, no matter where it goes, which is the recipient embryo in this case, it will still try to develop a brain cell. So eventually, we get the embryo with two heads. So that was very incredible and very fascinating. And the scientists were trying to understand how this process happens. How does a cell transplantation give rise to two heads? So what scientists have learned that each cell has the recipe in the form of mRNA, as Shannon talked earlier, that tells them who they are. So for example, if we have a brain cell, they have abundance of information that tells them that they want to be, that they need to be brain cell. So they just keep screaming, I want to be brain, I want to be brain. And this message is in the form of genetic code. So scientists really try to understand if those molecules are important for cell fate or not. So what they did, they took this particular message, see, I want to be brain message, they synthesized it in a tube, so made a synthetic mRNA message, and injected into these cells that are not supposed to be brain cells to see what will happen. So once the injection happened, now these cells that were injected are also trying to scream, I want to be brain, I want to be brain. So the question is, what do you think will happen to this embryo? How does this embryo, how would this embryo develop? Would we again get embryo with two heads, or it wouldn't have any effect? Or what do you think will happen? So interestingly, when scientists analyzed these particular injected embryos, again, they saw that embryos develop with two heads. So injection of this message gave rise to two heads. So, and this shows you the significance of the mRNA, the message that is important for each of our cells early on to determine who they are, to determine their fate, their identity, and which eventually helps them to give rise to different structures in the embryo. And this particular fate map, as I said, is created early on, and each cell positions themselves relative to each other in a way, like in a compass, so they give rise to the corresponding structure after several rounds of division. So what I told you so far, and I want you to walk away with today, is that cell fate is determined early on during development, as I showed by transplantation experiments and the injection experiments. And the second message I want you to get is that mRNA, this message, the cell recipe, specifies the cell identity and its fate. So what I mean by that is that the muscle cell, for example, the muscle cell has abundance of the message that tells it to become a muscle cell, while the brain cell has abundance of a message that tells it to become a brain cell. And it's all in a form of genetic code. So I hope it was clear, and I hope you learned something from this presentation. And in the next presentation, Hadass is going to talk about all the amazing things we learned about aging and all the amazing model organisms that scientists use to learn more about aging and how we can age in a better way, in a healthy way. And please welcome Hadass. Thank you.
Can you see my screen? All right. Hi, I'm Hadass. I'll be talking about aging today. And to talk about aging, I'll be focusing on three questions. These are, what is aging? How do we study it? And what can we do about it? And here I want to be clear that we are all aging all the time. This is a natural and essential process. And so our objective should always be to extend health span rather than lifespan. And what I mean by health span is that our goal is to extend the amount of time that we are healthy and functioning, and not just the amount of time that we are breathing. So to start, let's define aging. What is aging? Or at least what aging is to a cell. See, all our cells start from one cell that has the potential to create every type of cell in our body, like Shannon talked about, right? And then that cell divides and creates many others, most of which become one type of cell and stay that way for the rest of our lives, like these brainy cells eventually become, like Damir talked about. But I want to talk about what happens to our stem cells when we're full-grown adults, because stem cells within our tissues keep us healthy, but something happens to these stem cells when we age. And what happens to our stem cells is called cellular senescence. Once we are fully developed, a small number of our cells remain stem cells, here represented again by these orange circles, meaning they can continue to divide throughout our life and they replenish our cells that have been damaged. So for example, our muscle cells are mostly non-stem cells that make up the muscle tissue until those cells eventually die. And during our lifetime, they're replaced by new cells that have divided from the stem cell. So the stem cell is like this factory, always creating new cells. But eventually, these cell factories, these stem cells, stop dividing. They stop creating new cells, and this is called cellular senescence. So a big question is, why do stem cells eventually senesce? Can't we just have them keep divide, or can't we just have some more stem cells and live longer and healthier? All these questions are being asked by scientists and at this moment remain unanswered. So on a cellular level, cellular senescence is aging. But what is happening to those precious stem cells that is making them senesce? If you've read or heard a talk before about aging, you may have heard about telomeres. This hypothesis about telomeres is a hypothesis about aging or about why stem cells eventually senesce. So each stem cell and every cell in our body has all our genetic material. That is DNA compressed into these chromosomes, which are cookbooks in our analogy, right? Telomeres are these constructs that sit at the edge of chromosomes, and they protect our genetic material, sort of like these egglets, these little pieces of plastic protect our shoelaces. And sort of like eventually these can wear away and our shoelaces can unravel. With every cell division, the telomeres that are protecting the DNA get shorter and shorter, and eventually they're gone. At this point, cells senesce, and that is so as not to divide and pass on damaged genetic material, or at least this is the hypothesis. A lot of research has gone into testing this hypothesis, and some research has actually shown that telomere length is completely unrelated to mouse age, and that some human telomeres did not shorten over time at all. So we talked about what aging is, at least on the cellular level with senescence. And the next question I want to tackle is, how do we study aging? Especially because we're not sure this has anything to do with telomeres. So for this, I want to tell you about some amazingly interesting animals that we have lots to learn from and scientists are studying today. And I want to tell you just a little bit about genetics and what we can learn about aging through genetic studies. So some animals definitely have got down that slow, healthy aging much better than we do. And the best we can do is try to learn from them. Does anyone have a guess? You can write it in the chat, what the longest living animal known to us is. I see whale. Giant tortoise is a good turtle. Wow, we got a lot of turtles and tortoises and Galapagos tortoises, which are great guesses, and they do live very long, about 150 years. But actually the oldest animals known to us so far are 15,000-year-old Antarctic sponges. Antarctic sponges are not the answer to the question, who lives in a pineapple under the sea? And most of them also don't wear pants, but they have pretty much cracked the code to immortality, and scientists are studying how they do it. You might be asking yourself what we can possibly learn about our glorious selves from these porous animals that we use to wash our dishes. And so I will also present you with the longest living vertebrate, which means it's much closer to us evolutionarily. And this is no other than the Greenland shark, which lives to be about 400 years old. So it passes the tortoise there. Most hypotheses about both the Antarctic sponges and the Greenland shark and their ability to live so long have to do with the fact that they live in very cold temperatures. And living in very cold temperatures means they have very slow metabolism. And I'll explain this right now. So metabolism is our ability to turn molecules like those that we eat into energy. Energy is essential for growing and reproducing and interacting with our environments and generally living. So energy, in a way, is synonymous to life, right? We need it. And all processes in our body are molecules interacting with other molecules. So when we eat, there's a molecule in our stomach responsible for cutting up the molecules that we eat. But in the cold, that cutting molecule, it will cut, but it will cut very, very, very slowly. Cold equals slow metabolism, or in a way, slow living. Because metabolism is energy, which is life, which turns out could mean living longer. This connection between metabolism and lifespan was noticed a long time ago and posed as a theory called the rate of living theory. The rate of living theory and this was based on his observation that larger animals outlived smaller ones. So an elephant outlives a camel, which outlives a dog, and so on. And this is a pretty strong theory that holds true for many, but definitely not all cases. So just like temperature, body size affects metabolism. The bigger the animal, the slower the metabolism, the longer it lives. This brings me to our last very cool animal, the naked mole rat. This is the longest living mammal relative to its size, and that's very important. Most rodents of the same size live 2 or 3 years, where these handsome hairless rodents live around 20 to 30 years. So it makes a lot of sense to be studying the naked mole rat because sure, we know that cold temperatures and large body size slow metabolism, which elongates life. But humans are not becoming much bigger or adapting to life in Antarctic waters anytime soon. So studying an animal that is close to us evolutionarily, lives in a similar climate, and has a long life despite its very small body size means it probably has some trick up its sleeve that we have not discovered yet, and we're looking to discover as scientists. So scientists are studying these animals' telomeres and their stem cells and senescence, and they're studying their metabolism and behavior and genetics. In fact, scientists are studying all of these animals because there are many more questions to be answered and many ways to go about it. So while we're still on the topic of how we study aging, I want to give you a little idea of what geneticists like me can do in the lab to study aging. Geneticists study genes. So in our chromosome being a recipe book analogy, a gene is an individual recipe. And when I say expression of a gene can be turned up or down, I mean that we can have that recipe actually made. So we can have the cell make a lot of cookies or make none at all. And sometimes we can even change that recipe. There are many genes and many recipes, essentially, many of which have been associated with aging. I'm just going to talk about one as an example, but keep that in mind. So the one gene I want to give as an example is called FOXO3. This is just a name, so don't let it distract you from the point, which is that we can turn the expression of genes up and down and see what happens. So this is a very typical research strategy. And in the case of FOXO, when we completely shut it off, we get cellular senescence. This is great, right? We can just turn it all the way up and get stem cells that don't senesce and live forever, right? But when you do this, you turn up cell division and cell proliferation, and what you get a lot of the time is a tumor. And tumors are cells that divide too much and too often and result in an unwanted mass of cells. This happens when you deregulate the expression of many other genes as well, not just FOXO. So we want to find a way to stay somewhere here in the middle where we're not drowning in cookies, but also not going hungry. Which brings me to, what do we do about it? What do we do about aging? We've talked about what aging is with senescence and telomeres, and we've talked about how we study aging looking at cool animals and using genetics. And now what we can do about it, I want to, before I go into this, reiterate that aging isn't necessarily something bad that we should be fighting, but something we should be welcoming as long as we can stay healthy. So for what we can do about it, I want to share some really cool research about calorie restriction and about positive beliefs about aging. Calorie restriction does not mean anyone should starve themselves or be hungry ever, but restricting the amount any animal eats consistently prolongs their life. There is nearly nothing that is more solidly proven to help with healthy aging. Now, now that we've talked about metabolism, this really shouldn't come as a surprise, right? Because when we eat less, our body has less molecules to turn into energy, which means slow metabolism. And slow metabolism, with respect to the rate of living theory, means we are living slower, longer lives. And lastly, I want to tell you about studies by Professor Becca Levy, a professor of epidemiology and psychology at the Yale School of Public Health. And she has found that positive beliefs about aging may increase lifespan. She has dedicated her life to this research, and she's found some incredible connections between psychology and health in the context of aging specifically. And she's even shown that positive age beliefs protect against dementia, even if you have this high-risk gene. In this instance, she's referring to ApoE, which some variants of this gene can put people at a high risk of getting dementia. And when I say variants, I mean that we all have this gene called ApoE, but just like the recipes, there are many different recipes for chocolate chip cookies, right? So there are many different variants of this gene, and some variants put you at higher risk of getting dementia. And Professor Levy shows that positive beliefs about aging defy things that we think of as very, very solid, like genetics. So I want you all leaving this talk feeling positive about aging so that you age healthily and feel good for a very, very long time to come. Thank you.